Hai semua. For today's lesson, we will focus on the process of DNA replication. So what is DNA replication? It is the process in which new DNA molecules are synthesized. And we should know that this occurs during the S phase of the cell cycle. If we look at this chromosome over here, okay, we should know that this chromosome consists of one DNA molecule, and due to the process of DNA replication that occurs during the S phase in the cell cycle, this one DNA molecule now become two DNA molecules, okay, for one particular chromosome. And so if there are two homologous chromosomes in this diagram, we can see that each of the chromosome consists of two sister chromatids. And actually there are three models that can describe about the process of DNA replication. Number one will be the semi-conservative model. Number two will be the conservative model. And number three will be the dispersive model. What will be the difference between these three? Let's look at the conservative model first. In this conservative model, we start with a parental original DNA strand, which we know that one molecule of DNA consists of two DNA strands that are helical in shape. And so, in this conservative model, both of the parental DNA strands will remain together. They will not separate, and both of them will become templates to produce new DNA strands. And as a result, we can see that at the end of this replication process, one DNA molecule consists of two original parental DNA strands. Meanwhile, another DNA molecule over here consists of two newly formed DNA strands. Please compare with the second type. The second model, which is named as the semi-conservative model, shows that this original parental strand of DNA will separate. Okay, they will separate, and each of them will become templates to form new DNA strand. Okay, and as a result, at the end of this replication process, we can see that one DNA molecule over here consists of the original parental strand, which is dark blue in color over here, and also one new DNA strand. So it is a combination of old and new strands of DNA. The same goes for this other one molecule of DNA. But for the third model, the dispersive model, the original parental strand will become fragments. Okay, and each of the fragments will become template to form new DNA fragments. As a result, at the end of this replication process, one DNA molecule consists of a combination of the original old fragments and the newly formed fragments of DNA. Okay, the same goes for over here. So which one of these three will be the best one to describe the process of DNA replication? There was one experiment which was done by Messelson and Stahl, which proposed that the DNA replication occurs according to the semi-conservative model. And so, for our next discussion, we will follow the semi-conservative model to explain about how the DNA replication procedure occurs. But before that, we need to know enzymes and also some proteins that are involved in the process of DNA replication. Number one will be the DNA helicase. Number two will be the topoisomerase. Number three will be the single-strand binding protein, which is not an enzyme. It is just a type of protein. And number four will be the primase. Number five will be the DNA polymerase 3. Number six will be the DNA polymerase 1. And finally, the DNA ligase. And so, when we want to describe about the mechanism of the DNA replication, we must do this based on the semi-conservative model, in which we have to remember that the parental DNA strand must separate. And so this will require an enzyme named as the DNA helicase, in which the DNA helicase will catalyze the unwinding of the parental DNA strands. For example, the DNA helicase enzyme over here will catalyze the unwinding of the parental DNA strands at this particular spot, and this will form the replication fork, in which we can see that the original parental strands are separated over here. Usually, when the two DNA strands are separated over here, the two separated strands will have a tendency to recoil with each other, and this is known as the supercoiling of the strands. However, an enzyme will be involved. That will be the topoisomerase enzyme. The topoisomerase enzyme will prevent the supercoiling of the strands. This will ensure that the two separated strands of DNA will remain separated. And this needs to be stabilized further by the action of the single-strand binding proteins. The single-strand binding proteins are not enzymes, okay, and they are just proteins that will attach to the separated DNA strands in order to stabilize the strands until both of the parental DNA strands can be used as templates. Before a new DNA strand can be formed, a short RNA primer must be formed first, and this will be catalyzed by an enzyme known as the primase. So as you can see, the primase enzyme over here will join RNA nucleotides to form a short RNA primer, and then comes along another enzyme known as the DNA polymerase 3, which will then catalyze the formation of the new DNA strand. So as you can see, once the RNA primer has been formed by the primase, the DNA polymerase 3 enzyme will be able to form a new DNA strand based on the template of the original parental strand. And the new strand that are formed by the DNA polymerase 3 enzyme will be elongating in the direction of 5' to the 3'. The RNA primer, which is at the starting point of the newly made DNA strand, must be converted into DNA molecules. And so comes along another enzyme known as the DNA polymerase 1, which will catalyze the conversion of the RNA primer into DNA. So as you can see, the red RNA primer over here has been converted into another DNA strand. And finally, we can see that due to the action of the DNA polymerase 1 enzyme, the new DNA strand is now completely formed. According to the semi-conservative model, we must remember that both of the parental strands will become templates. So we must understand that the new DNA strands will be formed at both sides. Okay. However, on one side, the new DNA strand will be formed continuously from the direction of 5' to 3' towards the replication fork, and this is known as the leading strand. Meanwhile, on the other side, the lagging strand will be formed. This is the type of DNA strand which is formed discontinuously, even though it is also formed 5' to 3' away from the replication fork. What we see here will be the formation of short segments of DNA, which is known as the Okazaki fragments. The Okazaki fragments need to be joined with each other, and so comes along another enzyme known as the DNA ligase, which will catalyze the joining of the Okazaki fragments, and as a result, one long lagging strand will be produced finally. And when the replication completes, two DNA molecules will be formed, in which one DNA molecule consists of one original parental strand and also one new complementary strand over here. And so that is how one DNA molecule can become two DNA molecules through the process of DNA replication. I will see you guys again in the next lesson.